All right, we're going to now turn to coronavirus outbreak update. Dr. Ram Kapaka from CDC is going to provide a brief overview of the coronavirus outbreak on the phone. Dr. Kapaka, thank you very much for joining us, and the stage is yours.
Thank you very much. Hopefully you can all hear me. Let me know if there's a problem with the sound. Thanks a lot for the opportunity to speak to you this afternoon. I think Dr. Messonnier was originally slated to be on the agenda in this spot, but she's obviously otherwise engaged and had asked me to give you just a brief update on what we're working on with respect to the novel coronavirus outbreak. Hopefully you can see my slides. If we could go to the one right following the title. Just in terms of a quick overview of the presentation this afternoon, I'll give you a brief summary of the status of the response, and then we'll talk a little bit about coronaviruses, both just sort of the common coronaviruses, others that have caused problems in the recent past, and then an overview of COVID-19 disease. Next slide, and actually you can skip to slide number 4. Excuse me. I think most of you may have seen this in the news, but just so that we're clear on terminology, on February 11th, the World Health Organization announced an official name for the disease that's causing the coronavirus outbreak, and so that disease name is now COVID-19, and CDC will be updating our website and all other CDC materials to reflect the updated name. Next slide. As far as the current situation, as of February 12th, COVID-19 disease has been reported in 27 international locations in addition to the United States. And there are 14 infections that have been reported or had been reported in the U.S. in 6 states, and actually just a little earlier this morning, a 15th was reported in a 7th state. So it's actually 15 infections in the United States in 7 different states. There have been 2 instances of person-to-person spread reported and detected in the United States. Both, however, had occurred after close, prolonged contact with an individual who had recently returned from Wuhan, China, which is really the area most affected at present by this disease. The first death of an American citizen was announced in China a few days ago. The next slide just gives you a sort of a visual of both the parts of the world as well as the states of the United States that have reported COVID-19 disease. You can see that it affects multiple continents, primarily Asia, but certainly in North America and Australia as well, and Europe. And then, as I mentioned before, although this slide indicates 6 states, there's a 7th state, Texas, that had reported the 15th case earlier today. With respect to the CDC response, CDC established a COVID-19 incident management system on January 7th. While the immediate risk of this virus to the American public is believed to be low, right now CDC is nevertheless working with healthcare as well as industry partners to get a good understanding of issues such as the supply chain for PPE and to adjust response plans in the event that supplies become limited. CDC has also posted both guidance as well as strategies for prevention based on the current situation and the availability of PPE as we know it now. CDC has been and is expected to continue to upload the full genetic sequences of viruses from all identified U.S. Patients into GenBank as they become available, and this would presumably facilitate research into vaccine and other measures to respond to the outbreak. can skip to the next slide and the one after that. So if we move now to just a little bit background on these viruses, the virus causing the current outbreak belongs to a large family of viruses known as the Coronaviridae. They're known to cause respiratory illness. The first ones were isolated in the 1960s, and you can see from the picture at the right of the slide, they're named for the crown-like spikes on the surface that can be visualized with electron microscopy. There are 4 groupings: alpha, beta, gamma, and delta. And some of these viruses can spread between animals and people, and so this would be a zoonotic disease. Next slide. There are 7 human coronaviruses that have been described. 4 of them are common. They're commonly transmitted. They occur most commonly in children. And then there are the so-called other human coronaviruses that are listed at the bottom of the slide that have caused outbreaks both in the past and then, of course, the current outbreak. Next slide. So the common human coronaviruses are spread most commonly from infected persons through to others through respiratory droplets, so by coughing or sneezing. It generally requires close personal contact, such as touching or shaking hands, or touching an object or a surface that has the virus on it. These viruses typically or disease from these viruses typically occur in the fall and the winter, but certainly can occur year-round. As I mentioned before, young children are most likely to get infected. And it's believed that most people will be infected with at least one of these viruses at least once in their lifetime. Next slide. Excuse me. In terms of clinical presentation, these illnesses are usually, with the common human coronaviruses, are usually mild. They cause mild to moderate upper respiratory tract illnesses, similar to what we know as the common cold. Sometimes they could cause more severe disease, such as pneumonia or bronchitis. But this is more common in infants, older adults, people with underlying medical conditions that might weaken the immune system. Typical symptoms include runny nose, headache, cough, and the diagnosis is generally based on testing that's done on respiratory specimens or serum, and these tests are pretty readily available for the human coronaviruses. Next slide. Just from a historical context standpoint, there are 2 newer coronaviruses that have caused large outbreaks. Of course, in 2002, there was the SARS outbreak that emerged in China, where it was first recognized, and then in 2012, the so-called MERS outbreak that was recognized first in Saudi Arabia. In terms of the scope of these outbreaks, with SARS, there were 8,000 plus probable cases and 774 recorded deaths between 2002 and 2003, and there have been no known human cases of the SARS coronavirus since 2004. With respect to MERS, there were over 2,400 lab-confirmed cases and over 850 deaths. This outbreak began in 2012, and the numbers reported here are as of October 2019. Among the cases, there were 2 U.S. cases in 2014 among healthcare professionals. Next slide. Now we'll turn to an overview of COVID-19, which is a new correct name, I think, of the disease causing the current outbreak. COVID-19 was first identified in Wuhan, China in December 2019. And early on, many patients reported to have a link to a very large seafood and live animal market in Wuhan, China. Later on, patients didn't have the exposure to that or other animal markets, and that was taken as an indication of person-to-person spread in China. There has been travel-related exportation of cases, obviously given the regional spread that I showed you in the map earlier, and then the first U.S. case was reported on January 21st. CDC is continuing to report confirmed COVID-19 cases in the U.S. online, and I've given the website here. As I mentioned a few minutes ago, the 15th case was confirmed by CDC earlier this morning. Next slide. In terms of the spread of COVID-19, we're still trying to better understand the various potential routes of transmission, and that's an active focus of the epidemiological investigation of the CDC response, and certainly investigations similar to that are going on in other parts of the world. What we have been saying is largely based on what is known from the experience with other coronaviruses. Transmission is presumed to occur primarily through close person-to-person contact, and may occur when respiratory droplets are produced by an infected individual, and those droplets are produced when that person coughs or sneezes. It's possible that transmission could occur by touching a surface or object that has the virus on it, and then touching the mouth, nose, or eyes. But obviously, much of this is, as I said, based on prior experience, and the active investigation is ongoing to kind of confirm and understand this in greater detail. Next slide. With respect to symptoms, what has been observed so far is that symptoms can include fever, cough, shortness of breath. There is currently a wide range of illness severity being reported, from very mild cases, mild illness, to much more severe illness requiring hospitalization and intubation and mechanical ventilation. This infection with this virus can result in death. The estimated incubation period is 2 to 14 days, and complications, as with other respiratory viruses, can include pneumonia, respiratory failure, and multisystem organ failure. Next slide. In terms of prevention and treatment, I think what we want to make very clear is that everyday preventive actions for respiratory illnesses apply here. So what we seek to tell the public is that it's important to wash your hands often with soap and water for at least 20 seconds. If that's not possible, you can use an alcohol-based hand sanitizer with at least 60% alcohol. Avoid touching your eyes, nose, mouth with unwashed hands, and avoid close contact with people who are sick. If you are sick, you should stay home. If you're coughing and sneezing, cover your cough or sneeze with a tissue and then throw the tissue away. Clean and disinfect surfaces and objects frequently. And those are just basic principles that obviously we tell people for prevention of influenza, for other respiratory viruses, and they certainly apply in this case as well. With respect to treatment, there is no specific antiviral treatment that's currently licensed for COVID-19. Management really consists of supportive care to relieve symptoms and then managing the complications. NIH is actively working to develop therapeutics and vaccine approaches for treatment and spread. So just in terms of summary, COVID-19, there's an expanding active outbreak of respiratory illness affecting many parts of the world caused by a novel coronavirus. Most cases are still limited to mainland China. The virus is able to spread from person to person, but right now this virus doesn't appear to be spreading in the community in the United States. This is a rapidly evolving situation, and the risk assessment for Americans, which is currently low, may change. And the federal government is working very closely with partners from various sectors to respond to this important public health threat. So we have a few minutes probably for questions. If we don't get to your question and you want to still pose it, there's an email address which will come directly to the clinical team, and we can either respond to it or find somebody who can do so. So thank you very much, and let me know if there's any immediate questions I can address.
Thank you, Dr. Kopaka. Are there any questions from members of the committee? Dr. Meisner.
Thank you. There have been conflicting reports about the ability of asymptomatic individuals to spread COVID-19. Do you have any comments about that? And then secondly, do the commercially available hybridization assays that are used in many hospitals for picking up the 4 conventional coronaviruses, do you know if those assays cross-react with COVID-19?
Yeah, so the laboratory question, I think it'd probably be better for me to say that we can consult with the lab colleagues and get back with you because I think that's a pretty specific answer, and I'd like to make sure that the information we give you is 100% accurate. So we'll make a note of that and get that answer to that question to you. So with respect to the other issue about spread by asymptomatic individuals, I think that's a topic, again, of very active investigation. There are ongoing components to the epidemiological investigation that are hoping to shed some more definitive light on that, but we don't have a definitive answer yet. The hope is that the risk of transmission from people that are truly asymptomatic is minimal to none, but I think we would await more definitive data to be able to say that categorically.
Troy Knighton.
Hi there, Troy Knighton from the Department of Veterans Affairs. I was just wondering, how do you know antiviral treatment is not effective for this?
We don't know that it's not, but at the same time, we also don't have definitive evidence that there's a specific antiviral agent that is effective. I will say that there are some candidate drugs that may be effective, and there is work ongoing to develop trials to actually examine those questions so that if there is an agent that can be demonstrated to have true effect or efficacy, that it can then be, employed in these patients. So it's not correct to say that. It's neither correct to say that there's an agent that is effective, nor is it correct to say that agents universally are ineffective. We just don't have that information yet.
Larry Pickering.
Would you comment on any potential or real association between coronavirus and influenza?
Good, really important question. We don't have that. We don't really have any answer to that. So I'm going to maybe take a little liberty and interpret your question. What we wonder about is whether or not infection with one predisposes an individual to infection and perhaps worse disease with the other, or for that matter, whether co-infection is possible. So again, we have no data on that. It's certainly a question that is one that we'd like to get more information and an answer to, given that we're still in the midst of influenza season, and but we really don't have any information to be able to give you an answer. Other than to say, and it's preaching to the choir, but the one way to help make that question less important, perhaps, is for everybody to get vaccinated.
Dr. Kopaka, do you have time for one or two more questions?
Let's do one more.
Okay. Mary Ann Jackson.
Thank you very much. Because there are so many questions related to COVID-19, there's a high degree of anxiety among the American population. I just want to mention a downstream situation that I think we should be aware of, and that is there are some cybersecurity issues related to phishing scams that are going on now, targeting hospitals that identify the information coming from CDC-info National Contact Center, and it includes a link that says you can get information about numbers in your city. So I think we need to and we're talking about the internet and infectious diseases tomorrow, I think. This is just another one of those downstream consequences. Have you heard anything about that at the CDC?
I personally have not, but I'm glad you brought that up, and we'll speak with our communications colleagues and see what they've heard, or at least make them aware that this is something that others have become aware of. It's a very important issue, so thank you for mentioning it.
Thank you.
Dr. Kopacka, thank you so much for your time. We'll let you get back to your important work, and let's have a round of applause, please. Now, if I could have the members of our next...
We'll get the answer to that lab question back to you, hopefully soon.
Thank you very much.
Thanks.
Going to next have the members of our next panel please come to the stage. We're going to be talking about coronavirus vaccine development, opportunities, and potential pitfalls. We'll start with Dr. Alan Embry from NIH, who will provide an overview of the work NIH is doing in this area. He'll be followed by Dr. Ralph Barrett from the University of North Carolina, Chapel Hill, who'll discuss emerging coronaviruses, the outbreak, and the development of vaccines, followed by Dr. Mark Dennison from Vanderbilt, who will provide his thoughts on novel targets, antivirals, and strategies for multiple approaches to virus countermeasures and treatments. Thank you for joining us, gentlemen.
Good afternoon. Thank you for the opportunity to be here. I'm going to quickly talk with you about what NIAID is doing to respond to the severe acute respiratory syndrome coronavirus 2. That is the name of the virus leading to the disease now called COVID-19, as was discussed a minute ago, just to add to a little bit of confusion. Let's see, next slide. Okay, so I just wanted to start by briefly explaining that there is a broad USG-led effort that is being coordinated by the ASPR to oversee the USG response, and there are particular working groups that Robert Johnson from BARDA will be talking about later in therapeutics, vaccines, diagnostics, and clinical trials, the goal of which is to make sure that we have a very coordinated response, not only in prioritizing the most important medical countermeasures to potentially move forward, but also in resource allocation for those efforts. I will, of course, focus on NIAID's efforts, research and development efforts in vaccines to SARS-CoV-2 today. So NIAID is rapidly trying to accelerate research toward improving the understanding of SARS-CoV-2 and the associated disease. We're working with our extramural and intramural scientists to evaluate the potential cross-reactivity with existing SARS and MERS vaccine candidates, as well as monoclonal antibodies that already exist. Because of sequence divergence, particularly with MERS, we don't anticipate a high level of cross-reactivity with the new coronavirus, but we are going to be looking at some SARS candidates that may be available for cross-reactivity. NIAID scientists are also in the process, as I'm sure you've seen in the news, of developing SARS-CoV-2 specific vaccine candidates, and I'll touch on that in just a moment. And we're providing a number of resources to the scientific community to facilitate vaccine development. Currently, on the street, we have a couple of specific funding opportunities. Of course, we're encouraging our scientists to apply through our normal grant mechanisms, but specific to this coronavirus, the first title is too long to read, but this is a way for investigators to apply for emergency supplemental funding to their existing NIH grants, and it does not have to be within the scope of their current grant, to improve the understanding of the, I have trouble not saying novel coronavirus, SARS-CoV-2, development of medical countermeasures, and development of animal models to support medical countermeasure development and understanding of pathogenesis. Additionally, our 2020 Omnibus Broad Agency announcement now specifically solicits development of SARS-CoV-2 vaccines, therapeutics, and diagnostics. We're together with the CDC, we have been able to obtain a viral isolate from the first U.S. patient, and that viral isolate is now available through BEI resources. We're in contact with many investigators around the world who have also told us that they will be able to send us other viral isolates from different countries, and we will make those available as soon as possible. There is a joint USG sample sharing committee that will oversee the prioritization and distribution of patient samples as they become available to the U.S. government, to the highest priority developers and scientists. And many of our scientists are also creating a number of reagents, including molecular clones, plasmids, pseudoviruses, inactivated RNA, et cetera, to be able to share with the scientific community as soon as possible. So briefly, I thought I would touch on the vaccine you may have heard about in the news lately. The NIAID Vaccine Research Center SARS-CoV-2 vaccine candidate is a product of many years of research together with collaborators at the University of Texas Austin, Jason McClellan, and really understanding, taking some of the work that was, the foundations were laid with other viruses such as RSV to be able to optimize the SARS-CoV vaccine in a way that it will stabilize the structure and increase its immunogenicity. So in this picture, and I don't think this works, all you need to know is that through rational vaccine design, they have taken the outer spike sequence of the novel coronavirus protein and made stabilizing mutations in it. So normally that spike that sits on the outside of the virus is sort of like a mushroom, and it's fairly unstable. And the mushroom cap kind of pops open, and as you can see in the first panel with blue, this is the wild type spike, and you can see it extends out in its post-fusion state. But these stabilizing mutations that are added to the stem region of the protein keep it in its pre-fusion state, and that enables, that stability seems to enable a higher level of immunogenicity in animals. And so using that sequence, that optimizing sequence, the VRC has partnered with the company Moderna with funding from CEPI to rapidly advance this vaccine into GMP manufacturing and phase 1 clinical trials. So the GMP product is expected in March, and NIAID will support the first phase 1 study through our Infectious Disease Clinical Research Consortium, which is a new research clinical trial network of 9 clinical sites shown here. So because we've seen 3 coronavirus outbreaks in the past 2 decades, I think it's also important to be thinking longer term about how we deal with these coronaviruses that will continue to emerge from animal reservoirs. So I wanted to mention that efforts are already underway, not specifically for this outbreak, but to think about universal-like approaches in the same way that we're talking about them for influenza vaccines. And the Vaccine Research Center is already making strides in optimizing the antigen design of these kinds of vaccines to increase the potency and breadth, displaying multiple coronavirus spike antigens on nanoparticles, and using gene-based delivery approaches for rapid response. So I'll just finally end by reiterating that I think it is very clear now, and you'll hear more in the next 2 talks, that coronaviruses have obvious pandemic potential, and novel coronaviruses will continue to likely continually emerge from reservoirs. NIAID is rapidly advancing the development of SARS-CoV-2 vaccine candidates and will support investigators in developing their unique candidates as well. And in all these efforts, we think global collaboration and transparency across the U.S. government, as well as with our global stakeholders and partners, are critical. So with that, I'll stop and thank you.
Thank you very much. Dr. Barrick?
So I work at the University of North Carolina. It's a pleasure to be here to talk about the novel coronavirus, SARS 2.0. My slides were sent in before they changed the name, so you're going to get 2019 novel coronavirus. So the outline of the talk, I'm going to talk a little bit about emerging coronaviruses because I think it's important for the context of universal and the actual targets for the Group 2B vaccine design. So I'll talk about SARS, the pre-epidemic SARS-like bat viruses and drivers of this outbreak. I'll talk a little bit about the outbreak with a specific focus on the antigenic variation that exists in this virus as compared to other SARS-like viruses, and then talk a little bit about countermeasure work. And obviously, these are the respiratory viruses that emerged in the 21st century, shown on the slide. So if you think about coronaviruses, an important take-home message that these viruses have evolved rapidly over the last 800 years. In fact, the 4 human coronaviruses emerged anywhere between 200 and 800 years ago. The OC43 strain, the contemporary strain, was around the turn of the 20th century. There's a large number of emerging porcine and bovine coronaviruses that have jumped into the animal populations, followed by SARS, MERS, and the 2019 virus in the 21st century. So these viruses are accelerating in terms of their cross-species movement. The drivers of this on this slide include the drivers of coronavirus evolution, include the large size of the genome, the largest RNA viruses in nature. They have a relatively low mutation rate because of a fidelity complex, a replication complex that regulates fidelity, so that's unique among RNA viruses. This is regulated by a protein called NSP14 that Mark will talk about in more detail. But we actually think that this fidelity regulation can actually be altered based on environmental conditions, allowing the virus to change its mutation rate to allow for rapid adaptation. The viruses also undergo high rates of RNA recombination. 25% of the progeny in a mixed infection can be recombinants from the 2 parental viruses that enter the cell. And finally, the spike glycoprotein, which is shown here on the bottom of the slide, is quite plastic. It tolerates high rates of mutation, insertions, deletions. This can alter tropism, antigenicity, and lead to modular evolution, where component parts of either a protein or huge gene clusters can be moved between strains. So thinking about the SARS coronaviruses, the SARS coronaviruses that emerged in 2000, 2003 are shown in yellow here on this phylogenetic tree, and this includes the early, middle, late phase isolates, as well as strains from civets and raccoon dogs. But studies over the last 17 years have also identified a large cluster of SARS-like bat coronaviruses, which are shown in either light or dark pink. The light pink ones on the lower part of the slide have very low epidemic potential in that they can't use the human receptor for entry. The ones in dark pink at the top can use human receptors, and so these viruses can vary considerably, but can also replicate theoretically quite well in human cells, as well as use human receptors. And the 2019 virus clearly has its origins in bats. So SARS, the current paradigm is SARS was probably a bat virus related to this virus called WIV16, which is about 98% identical to SARS, that spilled into open markets where it infected humans and civets, setting up a transmission cycle with the civet intermediate host that caused the outbreak. So one of the questions in our lab was to ask the question, is SARS extinct? And what is the threat level of these other viruses? So there's a couple ways to do this. If you look at this phylogenetic tree, the little blue circle there, you can synthetically resurrect the viruses, let's say shown in red, or you can move the spike glycoproteins into a molecular clone and ask if those viruses can program infection of human cells. And the ones in the dark box at the top, which range from about 97, 98% identity to about 90% identity, can replicate fine on human cells, including primary human airway cells that have been taken from transplant recipients. And so these viruses are poised for cross-species transmission. So consequently, when you think of the SARS-like coronaviruses, you need to think of a large reservoir of heterogeneous related strains that fall into two categories, higher risk and lower risk viruses. So in 2003, SARS coronavirus emerged, causing 8,000 cases and about 700 deaths. Strains with 10% variation could emerge in the future. And in 2019, we have the novel coronavirus, which is about 22% different than SARS, which has now caused over 60,000 cases and 1,360 deaths globally. So when you think of the group 2B SARS-like viruses, especially in terms of vaccine design, you need to think of it like an encyclopedia where you have SARS and the 2019 novel coronavirus, which have natural isolates in bats, which have spilled into human populations. And then you have strains that are still circulating in bats that are high risk. And the variation here can go from 0 to 20, 25%. And that's your vaccine target if you're interested in long-term protection of human populations against this group of viruses. I should say that all the viruses on this list use human ACE2 for entry, grow on primary human airway cells, either in humans or in animal models, cause acute respiratory distress syndrome. They have age-related disease susceptibilities, so that either in humans or animals, they have a preferentially to cause more severe disease in aged animals. And many of the ones, as you're moving past 10%, can escape existing immune therapies. MERS is also similar in terms of its problems associated with vaccine design. There are two MERS-like strains. There's the MERS coronavirus of 2012, which is still circulating today. And there's a MERS 422 virus, which was identified in China, that's 35% different than MERS in its spike-like a protein, can use human DPP4 as a receptor, and grows fine in primary human airway cells. So think about these as large clusters of zoonotic viruses, which are the ultimate targets for vaccine design. So the old models for virus emergence was that you have viruses in a heterogeneous pool where a point mutation may occur during replication. This is random and rare. It spills into a secondary host or a human, requires additional rounds of mutation. So this is a mutation-driven process with several steps before you end up with an epidemic strain. What's really the likely model for emerging coronaviruses is that there are generalists in the heterogeneous pools that are capable of using human, bat, and other animal receptors. It may not be a mutation-driven process, at least initially in terms of spilling over into human or animal populations, and it may only require one or no mutations to drive an expanding outbreak. Okay, so 2019 emerged in early December, a cluster of cases associated with open markets. A coronavirus was identified in January 7th. The genome sequences were reported between January 9th and 11th, and person-to-person transmission was sort of confirmed by the Chinese around the 20th. I just put this up here with a few boxes to highlight, in terms of cases, not only mortality, but if you look at 8,243 critically ill individuals who probably require respiratory support for treatment. The RO is still undetermined for the new virus. It's either 1.4 to 2.2 or 2.5 to 3.8, depending on who you talk to. There's examples of super spreaders and examples of asymptomatic or mild spread that have been reported either in the literature or in social media. So where did this virus come from? This is the phylogenetic tree of the known group 2B coronaviruses, including SARS with one arrow. SHC014, which is a bat SARS-like virus, has another arrow. Where do these human strains, new 2019 strains, exist on the tree? Well, they represent a totally new clade of SARS-like viruses that differ by over 5,000 to 6,000 nucleotides from anything that we'd seen before. Its closest relative is a bat coronavirus called RATG13, which is about 96% identical. In other words, it has about 1,200 nucleotide changes as compared to the human strain. So this is a bat virus. It's a bat virus that was harbored in bats in China and has emerged in China. The genome organization of the virus shown here is very similar to SARS. SARS-CoV-2 is shown on the bottom. Green boxes indicate structural genes, brown boxes, replicase genes, gray boxes, accessory ORFs. The genome organizations are virtually identical. And this is just the green is the ACE2 receptor. The purple is the interface of the virus spike glycoprotein that grabs that receptor. And modeling and other studies have clearly shown that human ACE2 is the receptor for the virus. Now, I wanted to show the antigenic variation here because this is the target for vaccine design. SARS coronavirus is all the way here on the left, on your left. Middle and early phase isolates have minor mutations. Anything in green is an amino acid change. So you could see that the SARS epidemic had very little variation associated with it. The WIV16 virus, a bat virus, is the most likely progenitor for that 2002-2004 outbreak. And as you move through the pre-epidemic SARS-like viruses like WIV1 and SHCO14, more antigenic change. 2019 looks like a Christmas tree, as you can see in box. And HKU3 has about 30% antigenic variation in the spike. So that's the actual target for broad-based vaccines that you might want to have on a shelf to prevent future outbreaks. So the vaccine targets. Well, spike is the major target for neutralizing antibody. Neutralizing antibodies are protective against virus infection. Cellular immune responses can also be protective, but the majority of the protective response is associated with neutralizing antibodies. To produce broadly cross-reactive vaccines, you really need to target probably this cluster of high-risk SARS-like bat coronaviruses that range from SARS on one end to 2019 novel coronavirus on the other. The stem is more conserved than the head of the spike glycoprotein, so stem-based vaccines may provide broad-based breadth and protection against all of these viruses. However, the most potent neutralizing antibodies are to the globular head. In other words, it's the very top of the molecule. The bottom part of the molecule on that top line is the stem, okay? Top is more variable. Now, there's a variety of vaccine complications that have been identified either with SARS or MERS or other coronaviruses. One is that vaccine efficacy in aged populations using primates and mice as models is problematic. You can lose 15 to 20% of your neutralizing response just by aging a mouse, for example, 1 year. Heterologous group 2B SARS-like coronaviruses, even minor variation of 1 to 5% can reduce the neutralization activity by another 15 to 20-fold. So aged populations are a really high-risk group that you're going to have to think about in terms of management and boosting that immune response to provide long-term protection. They're also the ones that are most vulnerable. There's also a Th2 immune pathology that's been described with SARS-based vaccines. Both the nucleocapsid and the spike glycoprotein-based vaccines, if mixed with adjuvants that have a tendency to drive a Th2 immune response, like alum, can be really problematic, and you'll see large numbers of eosinophils and neutrophils and other inflammatory cells in the lungs of the animals, either with or without accompanying virus replication after challenge. There's some evidence of enhancing antibodies. I'm less sure about that phenotype, but it has been reported in the literature, so you need to keep that in mind. So finally, I just want to thank the people in my lab, my collaborators, people like Mark, who will be talking next. So thank you.
Thank you, Dr. Berry. Dr. Dennison?
Thank you. Good afternoon. I tried to uninvite myself, but Ann didn't let me, which was good because I said I'm really working on antivirals, and so. But as I thought about it, I thought this is really actually very important. I think when we're talking about emergencies, and I think it was Dr. Grunwald that brought this up earlier, that oftentimes with vaccines and approaches, we're chasing things, and we're chasing something that's new. Okay, so we need to be thinking about universal strategies. We also need to be thinking about comprehensive strategies. Those, when we think about something like what is going on right now with not only its biological, but its social implications, that we have multiple strategies for interventions, those that can come off the shelf, those that can be developmental. And I've become a fan of thinking about this in terms of antivirals because we believe that we have methodologies and approaches that can be used to really identify universal approaches to coronaviruses. And I want to describe a couple of those to you today. This work is nothing if not a tremendously group participation with NIAID support, with multiple university institutions and with partners in industry from the beginning. And it's been a 5-year or 6-year process that just unfortunately, serendipitously, really occurred with the publications and the availability of these compounds right as this began. So it's implementation from really a really unusual idea to actually use during an epidemic has occurred in less than 5 years time. Okay. And this just mentions the group again that I mentioned with the people. So what is the need for this? Well, I think this concept of broad diversity of bats has been covered very, very well that are outbreak ready. I think the failure of some antibodies to neutralize future zoonotic coronaviruses, and even during the SARS epidemic, well demonstrated from Ralph's lab and others, that antibodies made against the very earliest isolates of SARS weren't very good at neutralizing those even later in the epidemic. And so that these transitions can occur very quickly if there's adaptation over time. The time to develop vaccines, even though what is going on right now is certainly going to be the fastest in history, the most novel approaches and the fastest to humans of anything that's ever been done, it still can track well behind the trajectory of an epidemic. Universal vaccines across all coronaviruses will be difficult. They're variation across a broad range. And even if we're assuming it's just group 2B and 2C, the MERS-like and the SARS-like, for example, I think the challenges will be significant and they won't be overcome. I think they can be overcome, but I think they won't be overcome readily. And then finally, the potential for off-the-shelf use of highly conserved functions, targeting highly conserved functions. So what have our goals been? Our goals from the very beginning were to look at these issues to not even, so it's not a typical go, no-go strategy. I've learned drug development late in life as a second language, so I'm really enthusiastic, but I don't have depth about it. So we came to this with a different idea, is let's just use a go idea that says we want something that's going to target every coronavirus we test. So we set that bar high from the very beginning in terms of mammalian, human, bat-like, other groups of coronaviruses. Second, that these would have something that would have a high barrier to genetic resistance, that would have limited genetic paths, and that there would be a high fitness cost. These are obviously weird theoretical concepts to use as go, no-go concepts, but we felt that this is important if we're going to have anything that's going to have any lasting effect. Third, another unusual one, is that we thought it's important to think about extending the therapeutic window for prevention, amelioration, or treatment. This is other data that's come out of really animal models out of Ralph's lab and others that really talked about this issue that any of these compounds that have been tested, things that have been tested in vitro or in animals, the issue was the immunopathology of the disease that kicks in very quickly. And so you can do something to damage the virus, you can knock the virus way down, but if the damage is already done, the disease is already in place, then you're not going to have an effect on the disease. So additional things, obviously, would it be possible to use these as something that could be used as sort of a quasi-vaccine in a short term that you could use to decrease transmission in a really, like a cruise ship, for example? Or something, and particularly something, of course, that could be orally administered so that it would immediately be available to developing countries and environments that, in terms of the stability of a compound. So I'm not going to spend a lot of time in the replication. It's been talked about. For those of you, we're going a little bit into the biology. These are RNA viruses, plus strand viruses that replicate in the host cell cytoplasm. They usurp host membranes to form factories. Once they're inside, they're translated into proteins. Those proteins are then processed by viral proteases, and those proteins all assemble into replication and transcription complexes that then are used to copy the virus, to assemble new virus particles, and for those to be released from the cell. I could spend the whole time on this just talking about different topics, but I'd rather give you an example of something we've done rather than sort of broad themes, because I think it's more useful. As we talk about this, then there are areas within the coronavirus genome. It's like everything coronavirus, it's a little outrageous. These are the largest RNA genomes of any RNA virus family. They have the most proteins and the most complicated or complex replication complex organization. What's shown on the top is the coronavirus genome, sort of laid out 5' to 3', and the boxes all along it with numbers represent the non-structural proteins. Those are made essentially as one giant protein, a string of pearls, and then viral proteases chop those up into individual proteins that then work back together to copy the virus. And what you can see is when those that are numbered 4 through 16, They're variably conserved, and there's some core ones that are really highly conserved across all coronaviruses. And the particular region of those in the sort of the 7 through 14 or 15, 16 range, those are really the core replication proteins. And those functions are shown below. Particularly in number 12 is the RNA-dependent RNA polymerase, which is the target of the compounds I'll tell you about. Number 14 is the exoribonuclease, the proofreading exonuclease that I'll talk about a tiny bit. And then you can see there's multiple other proteins that all tend to work together as a very large multi-protein complex, which is pretty unprecedented among the RNA viruses. Somehow in their evolution, the coronaviruses have found a way to recapitulate essentially what looks like a DNA-based multi-protein replication enzyme. So I consider them non-anthropomorphically to be very smart. Okay. So this is what that looks like then. This is this complex that I mentioned. And what's important about this is that the colors are the same as above. And what's important about this complex, as you can see, is there's the polymerase, there's multiple proteins involved in RNA processivity, unwinding the RNA genome. But the critical one I'm going to mention is the relationship between the dark red and the blue one, the polymerase and the exonuclease, as well as a small orange cofactor there called NSP10. And the reason I want to do this is because coronaviruses are the first and only known virus order, or they're within the order Nidovirales. They're the first and only known virus group that encodes RNA-dependent RNA proofreading. And this was proposed in vitro systems, and it was first demonstrated in my lab and then collaborations with Ralph to demonstrate that the coronavirus have a unique capacity for regulating the replication fidelity. This has been a problem, and we've demonstrated it to be a problem. I'll show you right here. That this is an example with our murine virus that we're testing, where we demonstrated that coronaviruses have high native resistance to a whole bunch of nucleoside analogs. And then what we did is we genetically inactivated that proofreading exonuclease. So you can see in the black lines across the top that we could give increasing doses of ribavirin or 5-fluorouracil, and the virus just disregards it. It's clearly capable of preventing that from damaging the virus. But when we inactivate that enzyme, now the virus becomes profoundly sensitive to these compounds. And so our question was, is it even possible to identify nucleoside analogs that could overcome that proofreading function and target the virus? Because as many of you know, nucleoside analogs are probably the most common form of antivirals that are used. Acyclovir, AZT, I could go on and on about nucleoside analogs that are used to inhibit virus replication, but coronaviruses appear to be resistant, so we wanted to test that. Well, it turns out, I'll back up, it turns out surprisingly that I think not knowing a lot about drug development can sometimes be helpful in the sense that we identified 2 different groups to work with us, Gilead and then Emory University DRIVE program, that both had compounds that they said, there's been someone that's done some preliminary screening, suggests these might be useful. And they were both nucleoside analogs, and they were sent to us blindly. I didn't know what they were. And then we tested them. And so look at that black line across the top that's flat. And then these are the 2 compounds. On the left is remdesivir, the one that's currently in use now in China in trials. And on the right is what's called EIDD 1931, 2801, or NHC, in hydroxycytidine. And what you can see on the top is that if this is virus titer on the y-axis and concentration along the x-axis, but both of these compounds independently had profound activity against our model virus in vitro. And for those of you that care about this sort of thing, their EC50 values are very submicromolar. So that's a very, very active compound against these viruses. So this was a shock and a surprise to us, and led us to say that these must be doing something different that we don't understand. But while we're trying to understand that, we wanted to move forward with their development. So this, as we had identified this in vitro in Dr. Barrick's lab, then the work was quickly moved over to study these in human airway epithelial cells, in cell culture, and in animals. And what is shown here is the in vitro study showing that we're just about remdesivir now, that remdesivir inhibits multiple other human coronaviruses and potential zoonotic viruses. It's showing that with increased concentration that you get dramatic log reductions in virus replication. So this is meeting one of that odd criteria we set at the beginning, that this is showing very broad activity against a wide variety of viruses in different groups. And this shows it even a little bit more clearly. In blue are shown the beta coronaviruses. The top group, you can see the group that MERS is in, the 2Cs, the group that SARS is in, the new novel coronavirus, as well as many bat viruses. So all the viruses that have yellow in the box are ones that are inhibited by this compound. okay, including some of the alpha coronaviruses, human, murine coronavirus in the group 2As. And so every virus that's been tested has been profoundly sensitive to remdesivir. So what we wanted to do next then was say, can we make sure this drug is if it's being used, that there's going to be a genetic barrier to resistance, and if resistance occurs, that resistance is not going to make a virus that's better, basically. And so what we did is then we passaged the virus, and after doing this, we had a call with Gilead. I have 2 pieces of good news for you, I said. And they said, what's that? And my answer was, well, number one, it was really, really hard to get resistance in culture under the best conditions. We had to passage it 23 times and keep working at it to get resistance. And the other piece of good news is we got a little resistance, so we can see what the mechanism is. And so what we found out was that the virus, after passaging it 23 times, only had 2 mutations in it, and those occurred in the RNA-dependent RNA polymerase. And what we saw was that those were in the core of the polymerase, and that on the left side, on the graph, you can see the black line is wild type, with it showing its sensitivity, and the passage 23 virus is shown in purple, and that showed some significant resistance at lower concentrations that then could be overcome by just higher concentrations of the drug. So it was limited resistance, and we were able to begin to look at the mechanism. But what was more important than that was we wanted to know, okay, how do these viruses grow? So the next thing we did was tested them in direct competition, essentially taking the wild type and the passage 23 virus, essentially, and putting them in the same culture and seeing which virus wins. So it was a winner-take-all. So this is just showing on the left then what happens. Red is showing the resistant virus, and black the wild type virus. When those were combined, even when we gave 9 times more of the mutant virus, and then we passaged it multiple times in culture, the wild type virus always won. So this mutant virus, although it could grow, the resistant virus, although it could grow, it had a definite cost and fitness that didn't work well. And then work in the Barrick lab then showed on the right that this virus also has less illness associated with it, or less weight loss, and that animals recover more quickly. That would be the blue line and the red line, the high concentration of the virus, showing that this virus is more attenuated in animals as well. So these were, then the next thing that was done was then to actually test its efficacy in animals. And this work was done by Tim Sheehan, Amy Sims, and others in the Barrick lab, where basically remdesivir was given before or 1 day post-exposure, and at which point it really decreases disease in a mouse model of lethal SARS infection. It shows an improvement in weight or less weight loss, a better starting weight or at day 4 as well, decreased virus in the lungs, and then with prophylactic use, the animals with their score on the left or the far right is called the PIN-H score, and a higher score is worse disease. So the animals that were given this drug prophylactically or 1 day post-infection had much improvement over those animals that obtained the vehicle. Now, one point that's not on here is this idea I talked about, that when these compounds are given at a later stage, they'll always reduce virus titer, but they don't have a significant impact on virus disease. Okay? And so this is leaving us with this idea that we can get things that are really active drugs, that really work prophylactically, that really work early in disease, but when it's ARDS and it's settled in and you have all the complexities of that an antiviral alone is not going to rescue that particular disease. And that's not unusual to coronaviruses. That's likely to occur with all other organisms that are causing ARDS-like pictures. So what's the summary of this? So remdesivir, to summarize it, I also mentioned it's currently in trials in China. It is given IV, so it is an IV drug, which is one of its limitations. It potently inhibits multiple divergent coronaviruses. Its mechanism of action, which I didn't discuss, is that it's a chain terminator. Essentially, one molecule gets into an RNA chain, and that RNA chain is done. So it's a very potent inhibitor. There's a high barrier to resistance, which is difficult to achieve. Mutations are associated with fitness loss and attenuation. It's efficacious for prophylaxis, and it decreases disease and virus titer when administered early in infection. And you remember at the beginning I mentioned this other compound from Emory called N-hydroxycytidine. I wanted to use remdesivir as an example because it's now out and being tested in humans. But NHC is well along that path with a few steps to go. But I just wanted to point out this same information with that drug. It's orally administered, so it's orally bioavailable. I won't read through all the list again, but you can see the green check marks by all of the different things. as well. So this compound has really been passing our milestones in terms of its potential for use as well. It has a different mechanism of action, which is mutagenesis. It inserts mutations into the viral RNA genome, which impairs its replication and also impairs the fitness of viruses that come out. So it potentially could decrease virus spread because those viruses are less fit as they come out. So just back to finish up with this issue of countermeasures. We're really obviously working on direct-acting antivirals. We think that's very important for multiple reasons: treatment, prophylaxis, and potentially for decreasing transmission. Obviously, monoclonal antibodies are good at blocking infection and can act as passive immunization. Host-directed therapies, I think we need to think about because of these issues of immunopathology. And how do we really either make it so that it's possible for someone to survive while the direct-acting antiviral or monoclonal is working. And then finally, what we're really interested in, and this is the basis of the work that we're being supported by the NIH also, is to consider the issue of combinations of multiple direct-acting antivirals, even two nucleoside analogs like remdesivir and NHC, for example, direct-acting antivirals and monoclonal antibodies, and direct-acting antivirals and host-directed therapy to try to extend the therapeutic window and really give us more of an opportunity to help those who are ill, but also potentially to prevent infection in a more robust way. So I will stop with that, and thank you for your attention very much.
Thank all the members of the panel. Are any questions from members of the committee? You stunned us into silence. Okay. Well, you know, I think that this gives us at least some hope that despite the virulence and to take an anthropomorphic view, the smartness of these viruses, that there is some hope for the coronavirus changes that we've been facing. Thank you all very much for your presentations. Oh, I'm sorry, Mary Ann, go ahead.
Was the index case in the United States who was in Washington State treated, and do you have any data on that patient?
I don't have any data on the patient. I looked at the same information you did. The positive thing I took away from that was that there was the biochemicals were stable and there was no indication of a toxicity acutely, and I had the same data that you had. And as we all know, anecdotes can be used many different ways, right? I mean, the patient improved while on the drug, which is not helpful in an N of 1. But I think it was useful, and also since it had been used in the Ebola trials, remdesivir had been used safely in the Ebola trials. I think that's encouraging as well. I guess one other point I didn't make, which I really think is a really critical one, and people ask me, say, is the drug going to work or is the drug not going to work? I think that's the wrong question. The question is in what circumstances will the drug work? Because this drug clearly works in multiple animal models. Whether that'll translate directly, we don't know the answer to that question. But what we do know is if we design it to just treat those who are in ICU on a ventilator, that I think we might as well not design the trial, right? I think what we need to do is be sure that we're looking at variability, and we need to have virological indicators, biochemical indicators, as well as outcome indicators if we're going to really get information. It may not be the silver bullet, okay? Probably won't be just by itself, just that's the nature of these things, but that it puts us in a position to further evaluate it rather than calling it failed or succeeded, you know, winner take all.
Melody?
Thank you very much for the presentations. There was a lot of discussion about how these viruses are originating in bats. Can any of you explain maybe the transmission route that's taking place? Is it through droplets, inoculation from bites, ingestion of bats? How is this being transmitted to the humans?
Yes. Yes, I mean, I think, well, I think when I think you have population density, you have people going, Ralph can comment on this better, you can going into the caves, you know, you're having more and more exposures. There's wild game that may be able to support this, like civets were during the SARS epidemic, right? They're in these markets. And you're really having you may have a vertical transmission pathway of one animal on top of another animal on top of another one. And you have a virus that's a generalist and capable of using the same receptor or variations of that receptor in different animals. And then I think you can ping pong it back and forth to humans, which allows an amplification stage. So that's speculation of a model, but it's supportable by lots of pieces of individual data about these viruses and how they work.
Thanks, Mark. People that live and work around bat caves, bat hibernacula, many of them are seropositive, 1 to 3% are seropositive to these bat SARS-like group 2B coronaviruses. Now, how are they infected? Presumably, they may be collecting guano for fertilizer or for some other purpose. They may be harvesting bats for subsistence. But that's a very rich, laden environment with large numbers of bats, many of which can harbor up to 7 or 8 different coronaviruses in any given time. And so it's a very virus-laden environment that can allow for transmission to a person. Now, if you imagine a 2 to 14-day incubation period and people are traveling, they could easily take it to a mid-sized city where they could transmit it either to animals or to other people or to a larger-sized city to sort of spark an epidemic. But the fundamental question of how the virus gets from the bat to an outbreak situation in a community has not been worked out.
Len?
Thank you. This is probably a question for you, Dr. Embry. It may be too early for you to mention this, but I'm just curious about the types of clinical trials that may be planned, focusing in that the target population who seems to be most impacted by severity of disease are older adults, probably with comorbidities. So are we going to preferentially move quickly to that patient population? And then my understanding from some of the earlier work that was done with SARS vaccines that there were reports of antibody-dependent enhancement of infectivity and also eosinophilia. So are there particular safety parameters that are going to be looked at during vaccine development, or is it just too soon to tell?
So the initial vaccine that was purchased by the NIH, they purchased a doubly inactivated vaccine of SARS. That vaccine was mixed with alum. In both young and aged animals, if they are challenged with a lethal challenge virus, either homologous or heterologous, result in young animals, it will protect from infection but not immunopathology. Immunopathology is worse in a heterologous challenged animal. And in aged animals, it neither protects against, it may knock down replication of homologous, but it will not protect against heterologous. And Th2 immunopathology is rampant. Large numbers of neutrophils and eosinophils were in the lungs of the animals. It was primarily mediated by the nucleocapsid gene, but the spike gene also can induce a Th2 immune pathology. So this can be mitigated in part by using adjuvants that drive a more pro-inflammatory Th1 response. So that should be considered.
And if I can just add one point to that, you know, it's important to remember that these were animal studies. We really don't understand yet if these kinds of responses will be seen upon vaccination in humans. And of course, we are taking every safety measure to monitor that closely. But the vaccine in particular that I mentioned, developed by the VRC, was developed with this thought in mind. And the idea is that if you keep the spike in the pre-fusion state, you're most likely going to maximize the number of potent neutralizing antibodies that bind to the receptor binding domain, therefore neutralizing the virus before you have a sustained infection, and hopefully then mitigating the possibility of antibody enhancement.
I'd like to second that point as well. We have worked extensively with the VRC, sadly focusing on MERS coronavirus vaccines, and those vaccines induce a very robust neutralizing response to multiple domains. So I think it's a nice, safe platform to work from.
Tim?
Yeah, I just had some thoughts to share on Dr. Embry's mention that CEPI was involved in funding the program with Moderna. And I know we're going to hear from BARDA next, but I reminisced a little bit because in the February 2016
NVAC meeting, the late Adil Mahmoud stood on the stage and talked about the vision that he had shared in 2015 in a paper with Stanley Plotkin and Jeremy Farrar from Wellcome about this global organization that would be ready to do outbreaks much better than we did with Ebola. And it's great to see that happened. And I know that CEPI announced their first funding on January 23rd. I think they have 5 that they've announced so far. So it's great to see that come to fruition. And Adil Mahmoud, I worked with Adil Mahmoud at Merck, and he was not a patient man. So I think he would have really liked where this all went, but he would have said, much too slowly.
I just want to make one comment. Ralph's much older. I'm sorry, no, I'm older. We've been working on these viruses a long time, and shared grants and shared publications. And really, it was early Ralph's work back in the 1990s that predicted this potential for these viruses to move between species. And since the time of the SARS epidemic, particularly since MERS, we've been really focused on this. We're really committed to it. The viruses are, like I said, they really are showing a tremendous capacity. A couple final things. One, bats aren't our enemies. Okay, that's not the bat's fault. This is asymptomatic disease in these animals, and it's intrusion and changes in the environments, and they're very much at risk. So I know they don't need me to advocate for them, but I think they're an at-risk population. Number 2 is that we're very optimistic and hopeful about these approaches to vaccines and therapeutics. And we think it can be done much more quickly. And we think that the priorities have to be the continuity. So the fact that the U19 CEDARs allowed for us to do this kind of work over the last 5 years in a collaborative form is why we have 2 compounds that really we think are ones that could be tested now in humans. Whether they will succeed or not is a different question, but it can be done, and I think you can't put the mantle down. You have to keep going in terms of trying to think of strategies to target this group, because 3 times in 20 years from an evolutionary perspective is profound in terms of the risk associated with transmission to humans.
Thank you. Robert?
Thank you. I wonder if you have any comments about protease inhibitors or chloroquine or other potential agents.
Yeah, chloroquine is a broad alkalinization agent. It works against a lot of viruses at the level of entry. I mean, the idea of it working would be spectacular because it's, of course, most malaria is already chloroquine resistant, and so it's a compound that isn't really being used much, so it wouldn't theoretically have much implications in those areas. And a lot is known about its safety. So I think it's something that we want to go back and look at in combination with this kind of thing. Protease inhibitors have a tremendous, the coronaviruses have 2 or 3 proteases. I think this is probably SARS-like, so it probably has 2. And they're very critical to the virus and its replication. So theoretically, they'd be an excellent combination or single thing. Lots of times, ones that have been tested, we've tested, and others, we spent many years on them, they haven't crossed groups as well. So in other words, they'll work against the 2As, but there's enough structural difference. So as opposed to the polymerases, they look like they have more variability that's co-evolved within their group. That doesn't mean it can't. And there's one particular one that's been used in cats that's really widely used in China and other places right now. It's called GC376. That's been published because it converts this really lethal feline coronavirus called feline infectious peritonitis virus. It puts it in remission. And that's a chronic inflammatory immunopathogenic disease. So we think that they really need to be investigated. A combination of nucleoside analogs and protease inhibitors could potentially be a very great combination.
Thank you all very much for a very stimulating panel. We appreciate you. Our next presentation is from Dr. Robert Johnson. It's entitled Novel Coronaviruses, Medical Countermeasures, U.S. Task Force Efforts. Dr. Johnson's from the Biomedical Advanced Research and Development Authority. He'll update us on task force efforts to develop medical countermeasures for the 2019 novel coronavirus.
Okay. Great. Make sure I can work the remote here. So it's really appreciate the opportunity to have a chance to update all of you on our current efforts from the Medical Task Force area around development of medical countermeasures for the 2019 novel coronaviruses. I really want to talk about two areas today. First is the Medical Countermeasure Task Force that's been set up and kind of give an overview of what that is doing right now, and then spend the last couple of minutes talking about some of the things that BARD is interested in. So both of these things, both the Medical Countermeasure Task Force as well as some of the areas that BARD is looking at moving forward in, are alignment with the ASPR's mission around saving lives and protecting America from the 21st century health security threats. And really, when you think about what we're doing in this space, from my perspective, it comes down to this. As the previous speakers mentioned more than once, 16 years, third coronavirus outbreak, and we still have no licensed therapeutics and no licensed vaccines. And so the effort from my perspective is, what are we going to do to try to change that? And so one of the things that we have done is set up the Medical Countermeasure Task Force under the ASPR, and that Medical Countermeasure Task Force includes representatives from a majority of the agencies that are involved in various aspects of the response, and really focuses on those that are involved in medical countermeasure development. And so we have a wide array of representation, and the main goal, again, it's focused on medical countermeasures, all right? So how do we align our efforts across the U.S. government to try to reach that end goal and overcome this barrier that we have right now of no licensed vaccines and no licensed therapeutics? And so it's not just about sitting around and talking about things, it's really figuring out, what does that end-to-end solution look like? Who's going to handle what? We all know what our lanes are, but how are we handling that transition? How are we making sure that there's not a gap and we have a little bit of overlap there so we have a smooth transition? How can we utilize, from a synergistic perspective, if somebody has access to a particular model that others need access to, how do we make that happen so that we have the capacity that's needed to move forward? So what you see at this high level from the Medical Task Force is those types of discussions. But then what we have is we have the sub-working groups under that task force, and we've divided those into 4 categories. Not surprisingly, the therapeutics, the vaccines, the diagnostics, and the clinical trials. And each of those are focused at their level on this idea of looking across the entire spectrum of medical countermeasure development for their area, and what do we need to do now, and what do we need to do tomorrow, and what do we need to do next year, so that at the end of the day, we can get where we need to be. All right? And so we have those 4 prime subgroups, and these are not meetings where people come together and they talk about what they're doing. There's an active discussion around, what does the pipeline look like? How are we going to prioritize if we need to? Again, how do we make sure that everybody has access to the tools that they need? And as the data and the information is coming in on a daily basis about this virus, about what it looks like, about its pathology, and as we learn more about the models that are being developed, and as we get access to the virus and the more sequenced data, every day these groups are meeting and analyzing that data, assessing the plans that they have right now, and seeing how those plans need to adjust so that they can continue to move forward as quickly as possible. We also have 2 sub-working groups that really cross multiple of those working groups. One is around the animal models, obviously key for both therapeutic and vaccine development. Right now, of course, the big area of emphasis is what can we do in terms of just simply having some screening models, and then also beginning to look at what will our challenge models and our vaccination challenge models begin to look like. And then the second thing is around sample sharing. I think everybody's aware of the need for samples, the challenges of working with this particular bug right now with its BSL-3 category. So again, how can we get hold of the samples that we need, not just for testing our therapeutics and vaccines, but also for developing our diagnostics? How do we get hold of those samples? How do we make them widely available as quickly as possible? And so, again, these groups are meeting every day, and they discuss this data that's coming out, and they're figuring out solutions to move these products forward. And so when we think about end-to-end, what do we mean, right? Because everybody has, depending upon their viewpoint and their perspective, everybody has a different idea of what end-to-end means, right? Usually it means it's kind of where their work starts and their work ends. And what we mean from our perspective when we think about this task force, it really is getting from where we are today, getting down to not just licensing a product, but we have to then think about being able to push that product out, all right? If you have a vaccine that gets licensed, I mean, that's a huge goal. That's obviously something we want. But then we got to think about how do we get out enough of that vaccine as fast as possible to vaccinate the populations, all right? So again, we start on the left there. It's, yeah, your left as well, with the identification of the candidate medical countermeasures. All right, we start thinking about the animal models and preclinical that's needed. What's the regulatory pathway? From the previous speakers, you heard some very good conversations that have been going on around some of the some of the aspects that we have to think about as we develop products, some of the safety aspects, some of the efficacy aspects. Again, typical things for countermeasure development, but what's that regulatory pathway going to look like? We have to think about the phase 1, 2, 3 clinical trials. Again, how are we going to stagger that? And when we think about the clinical trials and the requirements, as we've seen over the last 20 years for every outbreak, right? We've all been through this, we've all seen this, and we know that it changes over time, right? The epidemiology of the outbreak will change over time. The needs and requirements will change over time. So we can put a plan in place today, and we will manage to that, and we will also recognize that that plan will continuously evolve, and that's especially true in the clinical trial arena. We have to start thinking about manufacturing scale-up early on. We can't wait until we have a vaccine that's licensed and then go back and say, oh, what about manufacturing? How are we going to get out enough doses to have a meaningful impact? I need to be thinking about that in parallel. We have, of course, the key milestone around licensure or clearance, depending on if you're a diagnostic or a vaccine or therapeutic, and then we actually go into that large-scale production. So it's this entire spectrum from the task force perspective that we're thinking about, and the groups are thinking about how to resolve and how to address. So I want to spend the last couple of minutes of this very brief overview just talking a little bit about some of the areas that BARDA is interested in exploring. So BARDA develops and makes available countermeasures by forming, really our mantra is forming unique public-private partnerships. So we partner and we move products forward towards licensure. When we think about the 2019 coronavirus medical countermeasure response strategy, we sort of lay out three primary principles. So one, we're looking to leverage our existing contracts, technologies, platforms, and capabilities to move forward as quickly as we can. The second is prioritizing development of MCMs based on public health impact and their probability of success. And then finally, thinking about this need for a domestic manufacturing base to produce whatever countermeasures are successfully brought over the line. We need to be able to ensure that we can produce the amount of product that's needed. So how do we look at this? We kind of divide it up into 4 quadrants. So the first is around vaccines. Focus right now for us is largely on the proven platforms that have large-scale vaccine capacity. Looking at multiple approaches. You know, one of the things that you heard from the previous speakers, and I think is a really important point, is that there's a lot we don't know about this virus. And really, looking at the past outbreaks, still a lot remains unknown about coronaviruses in general in these outbreaks. And so we all recognize, I think, the need to lay a broad base and not come in immediately on a very narrow base. We need to be looking at several different approaches to identify the ones that will work best as we move forward. Similarly, when we think about the therapeutics, again, thinking about platform-based, because the platform from our perspective, if they're successful early on, give us the opportunity to move forward the quickest into, again, not just license, but then having the facilities and the manufacturing base to move forward. Looking at repurposing therapeutics, again, common theme that a lot of groups are looking at, because again, it's that ability to have something available faster. And when we're thinking about responding to this outbreak, speed is a critical component of this. For the diagnostics, it's all about how early can we identify patients that are infected. That's really what it comes down to. So different approaches about how we might do that. And then finally, from a manufacturing, starting to think about now, what can we do with some of the flexible capabilities that we have access to, some of the facilities, so that once a successful product is identified, we can use some of these other capabilities to manufacture as much product as possible. So in closing, we have under the auspices of the task force, we do have a portal that's open for people that are interested, that have products, or not just products, but other approaches that they think that could help support this effort. They can put it into the portal. This is getting assessed and reviewed on a daily basis, so it's not one of those efforts where you kind of push it and you don't hear anything back. It's an active process that's reviewed every day, looking for the ideas that will help us to really move forward. In addition, we have under the BARDA Easy BAA and our regular BAA, we've opened it up for diagnostics, looking to develop and eventually clear diagnostics that will allow for a more rapid diagnosis of infection. So with that, I will conclude with those brief remarks and thank the audience for their attention.
John Douglas, are you on the phone? All right. Any comments or questions from members of the committee? Troy?
Hi, I'm Troy from the VA. Many of us worked on a task force last year for the president to sign that has to do with these. So I'm wondering if any funding or that executive order is of use yet.
Are you referring to the influenza executive order? Yeah. So that. So in terms of that executive order is really focused on pandemic influenza. But an important point to make is when we look at many of the requirements and the needs that support that EO, things like manufacturing base, speed, time, those flow almost exactly in parallel with this type of an outbreak. And so there is a lot of alignment in terms of needs around that. And so we do, when we think about it, we do go back to that. We go back and look at some of the influenza-related documents that you're very, very aware of that's out there about how we would respond in those types of things. So there is a lot of intertwining, and so support for one will be able to definitely support the other.
I think we all recognize there's lots of opportunity here if we're going to make progress and stop this sooner rather than later. Thank you very much for your presentation, John.
Thank you.
All right, we are at our afternoon break. We will break from now. We will start back promptly at 2:45.
Produced by the U.S. Department of Health and Human Services at taxpayer expense.